Male internal genital organs. The testis. The testes are a pair of male reproductive glands, which perform exocrine and endocrine functions at the same time. The exocrine function means producing male genital cells, spermatozoa, and the endocrine function means producing testosterone, male sexual hormone. The testes are situated in the scrotum and the left testis is situated a bit lower than the right one. The testis has an oval form, compact consistence and smooth surface. In the testis you see medial and a little bit protuberant lateral surfaces and also anterior and posterior borders, superior and inferior poles. The epididymis is situated on the top and behind the testis. Sometimes near the upper end of the testicle small appendage can be found. It is in appendix testis, which consists of rudimentary substance. The testis has a tunica albuginea, which is covered from the outside by internal, visceral, layer of the serous coat. From the internal surface of the posterior margin of the tunica albuginea a small roll-formed connective tissue formation juts into the parenchyma of the testis. The thin connective tissue septum of the testis move from the mediastinum to the parenchyma. They are fan-shaped and divide the parenchyma of the testis into lobules, 100,300 lobules. 1 or 2 convoluted seminiferous tubules are situated in each lobule. Near the mediastinum of the testis convoluted seminiferous tubules gradually become straight, join together in 2 or 3 and form short straight seminiferous tubules near the tops of the lobules. The straight seminiferous tubules flow into network of the testis which is situated in the thickness of the mediastinum of the testis. About 12-15 efferent ductules begin from the reed of the testis and flow into the duct of the epididymis. Convoluted seminiferous tubules are situated on one line with seminiferous epithelium and supporting cells, Sertoli cells, which are situated on the basement membrane. The cells of seminiferous epithelium which are at different stages of spermatogenesis from several layers of cells, stem cells, spermatogonia, spermatocytes, spermatids and spermatozoa can be found among. The spermatozoa are produced only in convoluted seminiferous tubules of the testis. All the rest canaliculi and ducts of the testis and the epididymis are the ways of excretion of spermatozoa. A liquid part of the sperm is presented by secretion of the seminal vesicles and the prostate. The supporting cells isolate the formed sexual cells from the toxic substances and different antigens, prevent the developing of immune reactions. The Sertoli cells can act like phagocytes. These cells also synthesize androgen-dependent protein which transfers the male sexual hormones to the spermatogenous cells. Besides that one more population of cells in the testis, they are interstitial endocrine cells, Leydig's cells. They are situated in the loose CT between convoluted seminiferous tubules around the blood capillaries too. The epididymis. It is in elongated form and situated along the upper and back margin of the testis. A wide upper part of the epididymis is called the head of the epididymis. It is directed downwards to the middle. More narrow part is the body of the epididymis. The body becomes narrow and turns into lower part tail of the epididymis. A little rudimentary appendage, appendix of epididymis, is the appendix of the epididymis and can be identified on the head of the epididymis. Sometimes in the region of the head and tail of the epididymis there are rudimentary formations, the defect ductules, which are presented by blindly finished tubules. The perididymis is flat white substance which is more expressed in childhood. The serous layer which covers the testis passes over to the epididymis. A recess between the testis and its epididymis which is covered with the serous layer is called the sinus of the epididymis. The efferent convoluted tubule of the testis form conical lobules, cones, of the epididymis. There are approximately 12-15 lobules in the epididymis. The tubule of each lobule opens into the long duct of the epididymis. The duct of epididymis in the tail region of the epididymis continues with the vas deferens. The epithelium of the vas deferens takes part in producing liquid which helps the sperm cell move through the seminal vesicles. Epididymis is a reservoir for storing the sperm cells where their biochemical structure becomes mature. When the spermatozoa leave the epididymis they are not fully mature and not ready for impregnation. 3. Ductus deferens. It is a paired tubular organ which is intended for the excretion of sperm cells. It starts from the duct of the epididymis, tail of epididymis, and finishes by confluence with the excretory duct of the seminal vesicle near the wall of the prostate region of the male. Urethra. 4 parts of the vas deferens. 1. The scrotal part situated behind the testis medial to its epididymis. 2. The funicular part ascends almost vertically, takes a medial position in the spermatic cord and reaches its superficial inguinal ring. 3. The inguinal part, it is situated in the inguinal canal of the spermatic cord and it ends near the deep inguinal ring. 4. The pelvic part goes backward and downward along the lateral wall of the pelvis and it has retroperitoneal location. Pelvic part goes between urethra and bladder, crosses through the urethra and then goes to the base of the bladder and reaches the base of the prostate. In the terminal portion of the vas deferens it becomes wide and spindle-shaped, forming the ampulla of the vas deferens. The lower part of the ampulla becomes narrow, then it enters the thickness of the prostate and joins the vas of the seminal gland. The wall of the vas deferens is thick. This wall consists of mucosa, muscular and the adventitia. The mucosa in the region of the ampulla forms recesses. From outside, the wall of the vas deferens is covered with the adventitia, which is evenly continuous with connective tissue surrounding the duct and the spermatic cord.